Hi you guys, this is a video to walk you through mini lesson number 5, I think it's 5, for Science 142, and you just follow along on this video and then write your answers down on the page that you have as your assignment in Google Docs. We're going to talk about protein synthesis and gene expression. The reason that I put AKA here is because that's also known as, and you can basically use these words interchangeably, so I like you to know both of them. We've talked a little bit about the central dogma of molecular biology before. In case you don't know what dogma means, it's like a creed to live by. It's like the important principles that matter in your life. And so in biology's life, this is the most important thing. I mean, biology isn't a person, but let's say it was. So when you're studying biology, this dogma is one of the most important things. And basically, what it describes is gene expression or protein synthesis. And we've said this before. You get DNA to RNA to protein, right? We've got DNA, it's going to get replicated, it's going to get transcribed into mRNA, and then we're going to build a protein. And that is really important because everything in your body is either made of proteins, made by proteins, or it functions because of proteins. So it's a really big deal to have proteins in your body. And the genes that are in DNA are how you get those proteins to be built. One thing, for example, if you don't have a gene for making this enzyme called lactase, then you're probably lactose intolerant. What happens is there's a sugar in your body called lactose if you drink milk, or there's a few other things that have lactose in them. And when you have those in your body needs to digest it. It's got to break down that sugar. And one of the things that breaks down sugar is going to be the enzyme lactase. So the lactose sugar is in your body, and if you don't have that right protein to break it down, then your body goes into all sorts of havoc and you get gas and diarrhea. But you can actually take that protein. So they make lactase pills called Lactaid, and if you take those, they'll help you digest the milk so you don't get all kinds of really uncomfortable gas. So this is important. When you have the directions, you can build the stuff in your body to do all the jobs that it has to do. So, the first part of this process is going to be called replication. And it's important to know which part of the cell cycle this is occurring in. So, replication isn't technically part of protein synthesis. A lot of times it's discussed at the same time. I would count it more as um, a, a cell cycle or a duplication topic, but since it always gets put with with the central dogma, we're going to go ahead and do that. DNA replication means copying the DNA. So you're just going to make a copy of it so that you have two. And when this happens in the cell cycle is during interphase. So remember, during the cell cycle, it's like mostly regular life. Then you get to the phase where a cell is duplicating, and then it goes back into regular life, and then it makes a copy, goes back into regular life. And so I don't know, this is specifically saying 8 to 10 hours, 6 to 8 hours, but I don't know exactly what cell this is talking about. Um, I just really liked this graph the best of all the ones that I could choose. So we're talking about DNA replication happening right here. The other two parts of this process, the RNA part and the protein synthesis part, those are literally happening all the time. All right, so let's zoom in a little bit on replication. Replication is going to happen inside of the nucleus of a eukaryotic cell. So, does replication occur in prokaryotes that don't have a nucleus? Yes, but we're not talking about them in this lecture. So when DNA copies itself, it happens in the nucleus. The DNA basically is not going to leave the nucleus unless a cell is dividing. And when it leaves the nucleus when the cell's dividing, remember it gets packed into those chromosomes so that they're easier not to get tangled and stuff. And that's how come, even though it's a really complicated molecule, for the most part, we copy it, and humans and all other organisms basically do a really good job of copying it without errors. Remember, there are errors, which are called mutations sometimes. So this first step happens in the nucleus, and what happens is that we're going to take those letters that make up DNA. So remember, the letters that make up DNA are called nucleotides, nitrogenous bases, base pairs, nucleic acids. You can call them a lot of different things, but they're basically always going to be listed as T, A, G, and C. And the reason I am saying them in that order is because T's are always going to match or fit with A's, and G's are always going to match and fit with C's. And you can actually see that they've made these shapes here to show you that that's the case. When a DNA gets copied, the molecule gets split open, the DNA molecule, by this enzyme called DNA polymerase, and it kind of unzips it, and then it copies one side at a time. And this is really cool because what you get are two copies at the end. But they're exactly like the parent copy. The only difference is that here they're showing the copied part in orange so that you can see that there's two copied sides. Hold on, my phone's ringing. All right, so they're just showing these color coding differences because that way you know which was the original. So like here's the original side of this, and this is the second original side of the original. But then you get the two copied sides. But because A's can only fit with T's and C's can only fit with G's, they get copied pretty well because that's the only way it can work. You cannot put a guanine and a adenine together. So you're going to get two daughter strands, and that's great because you need one copy for each of the daughter cells. So that's replication. Here's what it looks like when the enzyme is unzipping it. So see, you have an enzyme on this side represented by this blue thing. An enzyme is a type of protein. And then it basically just unzips it, and then it's going to add these nucleotides that are floating around in here, and it just fits them together with the strand of DNA. And so this is called a complementary copy. It's not an exact copy. If it was an exact copy, then you would copy a C with another C, right? But you can't do that in DNA because the C has to fit with the G. Otherwise, you can't put them together. They won't fit. So that's what complementary copy means. It means that it's a copy, but it's actually like the other side of the strand of DNA. So it's not copying exactly what's there. It's making a copy by pairing things. But since you're making two copies, you're going to pair both sides, and then you have two original copies, basically, or two daughter copies, but they're perfectly like the original, unless there's a mutation. All right, so we're going to practice this really quick in a, hold on, I have to figure out how to make this, no, escape. Okay, all right, I'm trying to go back now. I've gotten myself, okay, mixed up. So this is your worksheet, right, where you're going to be typing your answers. Hopefully you read all of these directions. Hopefully you've answered these questions already, because we are down here at 6 part B. So it says replicate the following strand. If you see this, and you're going to see it on your test, what it means is that you have to figure out what matches here. You have to make a copy of it. And you can't copy ATT because ATT will not fit with the original strand. So instead, you have to think about those pairs. Okay, well, A is going to fit with T, so my first letter is going to be T, and then T is going to fit with A, so my next one's going to be A. T, again, will fit with A. So my copy, my complementary copy of ATT is actually TAA, but that's still going to give me both sides of the DNA that I need. All right, let's do this next one together. What does C, or what does G go with? Hopefully you said it goes with C. What does C go with? It goes with G. So this copy would be CGG. I want you to do the other two on your own, and make sure you type this one in. I'm going to delete these. You type all of these in, but I'm just deleting it right now in the video. Okay, let's go back to our, okay, so replication. We just practiced replicating. That's what it's called as a noun form. Replication is a noun, and replicating is the verb of that. Sorry, I just said noun twice, but I meant to say verb. Okay, the next step of this, so step 2. Now this is technically going to be part of protein synthesis. DNA replication is only going to happen once in the cell's life, but transcription and translation are just going to be happening all the time. They just don't stop. Let me see if I can present from my current slide to make this a little bigger for you. Oh no. I guess we'll have to do this. Sorry guys. I just know that this shows up small on the screen, so I want to be able to zoom in a little bit. Okay, so transcription. This is still going to occur in the nucleus of the cell, but there's going to be two main differences between what we're making in this case, which is mRNA, and what we just made in the last step, which was DNA. So RNA, if you look, the most obvious difference between RNA and DNA, and you can see it down here in its coiled shape as well, is that RNA only has one strand, so it is not double-stranded. But your cell is not going to send the DNA out of its protective nuclear coating in order to make a protein every time, because that would be a huge mess and a big risk, and not very good for your cell. You might be messed up by that point if you were doing that all the time. So instead, a copy gets made of the DNA, but the copy is mRNA, and we call that RNA transcription. Specifically, we're making mRNA. All right, so let's go take a deeper look at this. So here's our picture of DNA and RNA. Sorry, it's cut off at the bottom, but that's the way Screencast-O-Matic works. And you can see here's the one strand of RNA. The other really important part that you need to remember of how RNA is different than DNA when we're talking about this mRNA is that C's are still going to go with G's, but we don't even have thymines. We don't even have T's in the case of RNA. So if you were looking at a string of letters that was supposed to be genetic information, you could tell if it was DNA or RNA. If it has U's, it's going to be RNA, and if it has T's, it's going to be DNA. So now for RNA, instead of A going with T, A is going to go with U. So whenever you want to put a T, you're going to put a U instead. Let's practice that. So going back to your little worksheet, which hopefully you answered all these questions, okay, we're going to do this with the same strand of DNA. So up here we had a TT that we copied, and it was TAA. Now, okay, we have A, and we want to put it with T because that's what we were just trained to do when it comes to DNA. But instead, we have to put it with a U in order to make this complementary copy. T is still going to go with A because A is still found in RNA. So we go like that. All right, so let's do this next one. It looks like I need to put some more spaces in between here so you guys can actually fit. Okay, so let's do GCC. Okay, so when we copy this, what does G go with? Still C. What does C go with? Still G. All right, now you do cat and GCA on your own. Back to our presentation. Okay. Go back to present. I don't want to present it from there. I just want to present it from right here. Maybe I'll just click on that. Oh, that didn't help. Okay. Present. Gonna have to get zooming through. Zoom, 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 zoom. Okay, the last step of this is called translation. And unlike the first two things we talked about, this is not going to occur in the nucleus. There's not enough room in there. It's going to occur out in the cytoplasm, more specifically out in the cytoplasm, and you need a ribosome to do it. So ribosomes, we've been saying, build proteins, and this is how they do it. There's also two other types of RNA that we haven't really talked about. There's rRNA and tRNA. The tRNA stands for transfer. Those ones are going to move the amino acids. And there's also rRNA, and it has several different jobs, but one of them is helping to match up the tRNA information with the mRNA information. We have to look really quick, though, at these little pieces up close. So this is kind of a zoomed in version. This is supposed to represent the nucleus. Here's the mRNA that comes out. It's meeting up with a ribosome out here in the cytoplasm. So let's talk about the details of this. Okay, the last picture we looked at didn't really show us the A's, the T's, or not T's, the A's, the U's, the G's, and the C's of the mRNA. But this one does. So it reads like C, G, U. G, U, G. So just like we had to make complementary copies, the tRNA has to fit with the message that's on the mRNA. We call that message a codon. So each set of 3 letters in the mRNA is a codon, and each of those corresponds to an amino acid. You'll remember from a previous lesson that amino acids are the monomer, so they're the little tiny piece of a polymer like a protein chain or what we call a polypeptide. So these amino acids are basically like these Legos that you put together into a bigger protein. And the way these match up is that there's an anticodon on the tRNA and there's a codon on the mRNA, and each one of them is carrying a little amino acid at the end. And when it comes, it matches up like a puzzle piece, and it's going to leave the right amino acid that corresponds to those 3 letters. So it's just going to drop it off, and the ribosome is going to help glue them together by bonding them with a chemical bond. And this is what you get at the end. So remember, we have our amino acids. Those are going to be our monomers. This would be a polymer. It's a chain of monomers, and we call it a polypeptide. And then this polypeptide, in order to officially be a protein, is going to fold, and it gets really complicated. This is the way we usually have, the way we show proteins when we're talking in biology. This is the way a chemist would draw it. They're concerned with all the different bonds. And then a physicist wants to know kind of what it actually looks like when it's all put together. So this is the most realistic depiction that we can have of a protein on a molecular level. Each one of those little spheres would represent one atom. So even though we're starting out with these little teeny pieces, these monomers of amino acids, when you put them together with the directions from a gene, what you end up getting is a protein that is like pretty darn complicated once it folds. Okay, we have to know how to use this information. So we've been talking about these codons, these 3-letter words. Well, how do we know which amino acid it's going to go with? We use what's called a codon chart. So remember, we have our codon down here and our anticodon, and the tRNA is going to match up with it. You can actually look at something like this, okay, AUC, which is a codon, a 3-letter word of mRNA, and you can use a codon wheel like this or a codon chart so that you know what amino acid it's going to code for. These little 3 sets of letters over here, these are shortened versions of the longer name for amino acids. So like LEU is leucine. This one's tyrosine, serine. There's also my favorite one, which is tryptophan, but I can't see it right now. Where is it? Tryptophan is TRP on this one. So is there a TRP on this one? Yeah, right there, tryptophan. Okay, so those are the names of the amino acids. That's all that's on the outside or on the inside of this chart. How do I know that AUC goes with ILL, which is isoleucine? Well, ILE. I go to the middle of my codon chart and I have A, okay, right here. And then I go to the second letter, so I'm going to move out from the center, which is U, so right here. And then I'm going to move out even one more into C, and that's going to get me to isoleucine right here. Now, you don't have to remember the words like isoleucine, like don't try to remember that. Okay, how do we use this other chart? Let's do GGA. I know it's going to be glycine, but let's figure out how to do it. So they have these as first position, second position, and then you're going to be looking in the chart box to find the last position. So let's start this with G. So G means that my answer is going to be in this bottom row. And then the next G is up here, so that means it's actually going to be in the last column. And actually, it doesn't matter what the third one is. If it's any of these letters, the amino acid is going to be glycine. So that's what I would put right here, glycine. So now I want you to try it with the codon wheel right here and see which one you get. So pause really quick, follow the directions from the inside out to say the amino acid. All right, here's the answer. So if we start in the middle with U right here, then we go to A right here, and then last we're going to have C, which is this one right here. So we're going to have TYR, which is tyrosine. Okay, now let's do it with the codon wheel on your own, and then I'll tell you the answer after. So pause, figure it out, and then I'll tell you what the amino acid is. So A is going to be our first position, so it's going to be in this row right here. Then we have to find the column U, so it's going to be right here. And G gets us to this last spot right here, so it's going to be methionine. All right, that is the presentation. We just need to practice it a little bit. We did just now. I'm going to practice once more on your worksheet, and then you guys are going to be able to just get this done and turn it in right here. You didn't even have to do any other parts of the lesson. Okay, I actually didn't do this ones yet on the lesson, so you'll see these right here. Oh no, it says translate the 4 codons. So this is your first codon, right? ATT. You can use the wheel or the chart. I find the wheel easier, so the one I'm going to do for you guys is the chart because I feel like that one's generally seen as harder. So you can open this up, and the first 3 letters we had in our DNA strand are A, or whatever we code this to. So remember, we just coded this when we transcribed it to UAA. So let's find UAA in our chart. Here's U on the top row. A would be in this block right here. And what was the last one? I forgot already. I think it was UAA. Yeah. Okay, A is the second to last one. So it's actually going to be, hold on. Wait, I already forgot what it is. I need to be able to see it. UAA. Okay, so UAA. It's going to be a stop codon. So that literally just tells it to stop. Like, don't do it. Okay, I hope that, and so what I would do is I would write that on this chart down here. I'll say stop. So you guys are going to do the other 3 right there, answer your essential question, and you guys are good to go for this mini lesson. Thanks, guys. Have a good day.